Hai, Assalamualaikum. My name is Nurfira Karnisa along with my member group Muhammad Nabil Alif, Isfahan Tasnim and Wan Shazza Mazwin will be presenting about innovation in zoology in this video. So, as we know, in zoology, innovation plays a vital role in expanding our understanding of animals and strengthening conservation efforts. Modern tools such as camera traps, drones, and remote sensing devices have transformed how we study wildlife. Camera traps reveal natural behaviors that humans might miss. Drones allow researchers to explore hard-to-reach or risky areas. And remote sensing track changes in habitats, migration, and population trends over time. These technologies help scientists collect accurate, long-term data with minimal disturbance to animals. As a result, researchers can make quicker and more informed decisions to protect species and maintain healthy ecosystems.
Let's move on to the problem statement. First is, rapid environmental change and habitat destruction are causing a sharp decline in animal population. Second, traditional zoology methods are too slow or limited to monitor and protect species effectively. Third, many species are become in danger or extinct before they can be studied or conserved. And lastly, lack of advanced technology reduce the accuracy of data and animal behavior, migration, genetic, and ecosystem. Without innovation, zoologists will struggle to preserve ecological balance and support future scientific discoveries.
For the innovation part, there are two examples of innovation in zoology that I choose. The first one is automated species identification and the second one is animal cloning. So what is automated species identification? Automated species identification refers to the use of artificial intelligence, machine learning and deep learning to automatically recognize animal species from image, audio or video. This technology is becoming one of the biggest breakthroughs in zoological research because it allows scientists to analyze huge data sets collected from camera traps, drones and acoustic sensors quickly and accurately without relying fully on manual identification. So why it is needed? It is because traditional species identification is slow, error-prone, costly and limited. So with the help of AI, it solves this challenge by processing large volume of data in minutes instead of months, reducing human mistakes, detecting species in poor lighting, partial visibility or complex habitats, and helping researchers monitor wildlife at large spatial scales and in real time. This directly accelerates biodiversity monitoring, conservation planning, and ecological research. How automated species identification works? There are three methods of this AI used in zoological. So the first one is convolutional neural network (CNN). It is the most common deep learning method. CNN learn visual patterns such as shape, texture, color, and body structure to classify species from image. It is used mainly on camera trap image trained using thousands to millions of label images. CNN also achieve high accuracy for large mammals, reptiles, birds, amphibians, and even insects. The second method is transfer learning. Transfer learning using models pre-trained on large image datasets like ImageNet, then fine-tuning them on wildlife datasets. It is useful when wildlife images are small in number, and it is also improved accuracy for rare or hard to photograph species. And the last one is AI-powered drone. Drones are equipped with cameras feed image or videos into AI models to count animals, also used to map their location and identify species in real time across large landscapes. Next, we go to animal cloning. So what is animal cloning? Cloning is the process of creating a genetic copy of an organism whose DNA is essentially identical to that of another. In animal cloning, scientists produce a new animal that has the same genetic makeup as a donor animal. Cloning does not require the usual sexual reproduction, which is a fusion of sperm and egg. Instead, through biotechnological methods, the cloned individual develops from non-reproductive cells, somatic cells of the donor. In nature, cloning loosely occurs, for example, in identical twins or asexual reproduction in plants and bacteria, but artificial cloning a full genetically identical copy of an animal that normally reproduces sexually is a human-driven technique to replicate an existing individual. There are several motivations why we need animal cloning. One of the main reasons are livestock and agriculture improvement. Cloning can be used to reproduce animals with desirable traits, allowing farmers to duplicate high-performing individuals rather than relying on uncertain breeding. The next reason is medical and pharmaceutical use. Cloned animals can be engineered or already naturally possess traits useful for medicines. And lastly, conservation or biodiversity application. In theory, cloning could help preserve or restore endangered species by reproducing individuals to boost population or maintain genetic lines. Thus, animal cloning is not purely a science experiment, but has a practical and economic motivation across agriculture, how animal cloning conservation, and there are two methods used, and the first one is embryo splitting or artificial twinning. An early stage embryo, already fertilized, is split manually into two or more parts. Each part develops separately into an individual embryo, then into separate identical animals. This essentially mimics the natural identical twin process. It can rapidly produce multiple genetically identical animals useful in livestock to multiply desirable traits. The second method used is somatic cell nuclear transfer (SCNT). This is the method used to produce famous clone. The first process is enucleation. A donor egg or site is taken, and its nucleus containing its DNA is removed, creating an empty egg. Next is nucleus extraction. From the animal to be cloned, the donor, a somatic cell, for example a skin cell or other non-reproductive cell, is taken. Its nucleus, which contains the full set of DNA, is extracted. Next process is nucleus transfer. The donor nucleus is inserted into the enucleated egg. Now the egg contains the full genetic material of the donor animal. The following process is stimulation and division. The reconstructed egg is stimulated, often by a small electric pulse, to start dividing and develop as an embryo. And the last process is embryo implantation. Once the embryo has reached a proper stage, it is implanted into a surrogate mother's uterus, where it can develop to term and be born naturally. If everything goes well, the result is a newborn animal whose nuclear DNA is bearing mitochondrial DNA differences identical to that of the donor. Because SCNT demonstrated that even adult somatic cells, not just embryonic cells, can be reprogrammed to develop into a whole organism, it opened the door to cloning fully grown animals, not just embryos.
Use AI and technology for recognized animals. For benefit, number one, we have faster identification. That system can recognize species quickly without needing human to check one by one. And second, more accurate result. Reduce human error using AI, image analysis, and genetic tools. And third, help monitor biodiversity, useful for tracking animals in forests, ocean, and protected area. For challenges, requires high quality data. Poor image or sound recording are reduced accuracy. And second, limited database. AI may not recognize a rare species with a little training data. And lastly, requires skilled workers. Requires expert in AI, coding, and zoology. Animal cloning also have benefit too. Number one, increase livestock production. Cloning can help farmers to produce more animals to meet global food demand. Second, stem cell advance. Cloning help create stem cells that may be used to treat many human diseases. And third, pet cloning. Allow people to clone a beloved pet that had died. For challenges, low success rate. Many cloned embryos fail to develop or survive. And second, high cost. Cloning products are extremely expensive. And lastly, environmental risk. Adding cloned animals into the wild may disturb natural population.
To wrap up, zoological advancement not only expand our scientific knowledge but also strengthen the way we study and protect animals. Moving forward, we need to keep adapting and exploring new tools and strategies to stay ahead. In the end, the future of zoology depends on continuous innovation so we can safeguard wildlife and maintain healthy and stable ecosystem. That's all from us. Thank you.